morning, everyone. Thank you so much for inviting me to speak today. So as Benny said, I'm going to talk about dust composition and effects in the American West. So really there's two main motivations for why I started this research project. And the first was I was really interested in how dust deposition influences ecosystems. So back 15 years ago now, when I started my PhD, I had heard a lot about how dust can impoverish ecosystems that are being eroded. It can remove a lot of the nutrients and the fine grain organic matter from soils. So I was really interested in where is that going and is it doing anything to where it's going? And I had heard anecdotally a lot of people talking about very recent increases in dust emissions and dust deposition, but at the time we didn't really have a lot of data to suggest that that was happening. And so we were able to show at that time that dust was increasing through that time period. And we were also able to correlate the composition of atmospheric deposition, both nitrogen and phosphorus, to lake ecosystems. And we could show that there was a correlation between the composition of dust and nitrogen deposition in both dry and wet deposition and what we saw referenced in sensitive lake ecosystems. And then we also saw through time that there was parallels between the amount of dust being emitted, the amount of dust being deposited, as well as the nutrient concentrations in those lakes.
Janice, we cannot see the slides.
Oh, really?
Yes.
So this worked, the sharing worked before.
Let's try it again.
Okay, can you see this?
No.
What's happening? Okay.
Let's do the stop sharing and we'll do it again. We know it worked.
Okay. Let's try this again.
No. We can see them now.
This is working?
Yes.
It works.
We can.
Okay, great. Sorry about that. Okay, so I was talking about how I was really interested in how dust might be influencing ecosystems, and particularly interested in freshwater ecosystems. And so we looked at dust deposition through time in places like the Intermountain West and the Northwest of the United States, and we saw increases. We also saw this parallel between the composition of atmospheric deposition and lake water chemistry over space. And we also saw parallels in dust deposition and freshwater chemistry through time. So this is data from a sediment core comparing TSP emissions from oil and gas activities with dust deposition in the downwind area, as well as phosphorus concentrations in the lake water. And then broadly across the United States, there is this suggestion that our really oligotrophic, low nutrient freshwater systems are disappearing in really remote locations, and one of the drivers might in fact be dust. And so all of this data is really compelling, and it provides a really interesting suggestion that the increased dust flux might be leading to increases in the trophic status of freshwater systems and changing their ecology. But it's really just based on correlations in space and time. It was really difficult at the time to collect enough materials so that we could do experiments to really understand how dust might be changing freshwater ecosystems. So this backdrop of this question motivated a lot of the work that I started doing as a faculty member. That and the fact that I think every single one of my PhD papers was rejected with the reviewer saying that dust phosphorus is not bioavailable and therefore this work is irrelevant. The other thing I noticed at that time was that I was really trying to get a lot of data on what we knew about atmospheric particulate composition and how that was changing through space and time and what it was composed of. But one of the challenges I routinely encounters was that we really only monitor particles less than 10 microns. And there's really good reasons to do that. But if you look at the graph on the right, this is data that was collected from a colleague in mountains in China. And this mountain range is impacted by both pollution from major urban centers as well as frequent dust events. And you can see this bimodal pattern. The pattern that peaks around 10 microns is when they have air masses come in from major urban centers. And then this broader peak is when they have dust events bringing material into that area. And so I've highlighted on this graph the data that we routinely collect and understand, as well as the samples that aren't routinely collected or measured or monitored. And of course, a lot of us do collect this type of data, but generally it has been infrequent in space or time. And so this fraction that we're not measuring means that we have this really big knowledge gap as to what is in the atmosphere, how much of it is moving around, and what it's composed of and what potentially it might be doing to sensitive ecosystems. And in my case, I'm really interested in aquatic ecosystems. So I thought one way that we could potentially fill this gap is to capitalize on an existing network. So I collaborated with the National Atmospheric Deposition Program to use their monitoring program. And if you're unfamiliar with them, they monitor rain chemistry. They collect samples every Tuesday, so once a week, at over 300 sites around the country. And this is what a typical station looks like. I'm not sure if you can see my cursor, but this is their wet and dry deposition sampler. They had not been using the dry side of the sampler. And the way this works is that there's a bucket here. It's open to the atmosphere under dry conditions. And then when it starts to rain, it triggers the sensor that moves the lid from the wet bucket over the dry bucket, and precipitation is captured in the precipitation bucket. And there's a heater on that sensor, so when it stops raining, it evaporates the water on the sensor and it moves the lid back over. So in this way, both dry and wet deposition can be captured independently. And so we developed a sampler that can fit inside the dry bucket, and it's composed of a series of screens that act to prevent any contamination, like bugs that might die in the sampler, from getting into the sample. And it also acts as a wind baffle, so it prevents any of that fine-grained material being re-excavated out of the sampler. And it lands on a glass plate that we then use a ceramic razor blade to capture what has fallen on that plate. And so this allowed us to measure the amount of dry deposition. We do this monthly across many different sites. We have just over 30 sites at the moment. And it also gives us a lot of material that we can do experiments with. And so I'm just going to preface this that we've been doing this since 2017. We have so much data. It's honestly kind of overwhelming, and I obviously cannot show everything that we have collected. I've put a lot of slides at the end of the talk, trying to anticipate questions that you guys might have, and data that I might be able to show unrelated to what I'm going to talk about today. But also, I would like to invite anyone who thinks they can value add to this project, who wants to collaborate. I'm very open to any of that moving forward. Okay. So from 2017 to 2022, I collated the data, our monthly data, into annual deposition rates. So you can see there's quite a range across our different sites. In general, the deposition rates ranged from about 3 to 15 grams per meter square per year. There's a handful of sites that show much greater deposition rates. These ones I've highlighted here. The ones in orange is because they're capturing a lot of the saltation flux. These samplers are actually only 1.5 meters off the ground. And so they're capturing a lot of very local material. And this green site is actually near the Boulder Urban Center, so it's capturing a lot of urban dust. But in general, we see a range between 14 and 15 grams per meter square per year. And each site has kind of different seasonal fluctuations, but this is what one site looks like. This is Colorado 10. This is in the central western part of the state. For the initial 1.5 years of the program, we also captured the particle deposition that occurred with wet fall, so particles that came in with rain. We didn't continue doing this because it was quite expensive, but it did give us an indication of how much was falling out dry versus how much was falling out wet at our different sites. Overall, between 40 to 90% of the mass was coming in with dry fall. But at some sites, or one site in particular at Colorado 98, the bulk of the material is coming in with wet precipitation. So looking at these samples under the microscopes, these are some different magnifications. I have a very nice microscope to look at these samples in detail. As expected, we see a lot of amorphous organic matter and minerals, so it looks very similar to soils under the microscope. We see a lot of things that are traditionally considered part of dust, like pollen and charcoal, insect parts and spores. Some of this certainly comes with the soil, but a lot of it is generated outside of the soil environment. And then we also see a lot of things that we didn't anticipate finding, like plastics and tire wear particles. So quite a range of material in our samples. By particle count, overall, about half the material was organic, 46% mineral, and about 4% polymeric. Now, I'm really interested in the ecological impacts of dust deposition or particle mass deposition. So I'm really interested in everything that's falling out of the sky and how that might potentially impact an aquatic ecosystem, so long as it's coming from outside of the catchment. So I also looked at color just for fun. I'm not sure what I'm going to do with this data, but it gives us a nice indication about how the composition of deposition changes through space and through time. And so we can see some interesting things pop out of this. For example, when the fire happened in the Sierra Mountains, this sampler is in California. We saw a lot of ashfall deposition. We could see the color got very dark during that period. We also see these reddish colored dust at more northern sites in Colorado sometimes appear. And then sometimes we see really interesting things like algae in our samples. So as I mentioned, I was really interested in phosphorus deposition and the impacts that might have on freshwater systems. And so I wanted to understand how bioavailable the phosphorus is in the dust samples that we're collecting. And so we did a sequential extraction to try to understand that. And we did a 5-part extraction. So the first fraction we looked at is the readily exchangeable inorganic phosphorus fraction. So if we add a weak salt and see what it can immediately leach out of the sample, we consider this to be readily bioavailable. So this is immediately available to any organisms, algae or bacterioplankton in a freshwater system. And then we looked at 2 different fractions where we could look at what is the labile organic fraction. We could isolate that. And that's the fraction that's available after some period of time. So after organisms have had a chance to try to break down that material to access that phosphorus. And then we looked at 2 fractions that are not really bioavailable under the time scales that we're interested in. This is the calcium-associated phosphorus, as well as the residual recalcitrant phosphorus. So in terms of the actual rates of total phosphorus deposition, so all of those fractions together, it ranged from 4 to 60 milligrams per meter square per year. And then breaking that down into how much is bioavailable, we see that anywhere between 37 and 70% of that total phosphorus is bioavailable. So what's shown here on the graph is the seasonal distribution between how bioavailable that phosphorus is. Broadly, the winter versus the summer, and how that changes over space. And if we look at what that looks like at just one site, this is data from northeastern Utah. You can see that a very small fraction is immediately bioavailable, but a much larger fraction of this organic labile phosphorus is what we see in this particular site. We also looked at the leachable ammonium and nitrates off the dust samples. And here on the upper left, I'm showing the gradient of what we were able to remove from those samples, just with also a weak salt leach. And then we also looked at the organic nitrogen and the organic carbon fraction. You can see a really big bullseye over California, and I believe that this bullseye is mostly influenced by a particular fire that happened in one of the samples where we collected a lot of ash in that one deposition month that is kind of overwhelming that site. But it's an interesting data point. And then looking at soluble reactive phosphorus versus organic phosphorus, we also see some interesting trends in space. And again, we see the effect of that fire event. I also looked at the ratio of nitrogen to phosphorus. This is an interesting thing to look at when you're considering the effects on aquatic communities. If we consider the dry flux of nitrogen as well as the wet flux of nitrogen against phosphorus, we can see that these ratios are well above Redfield. We consider only the dry flux of nitrogen versus phosphorus. That phosphorus fraction within the dust is overwhelming that ratio, and we get all of our values are well below Redfield. But if we look at just the easily leachable fraction, so that immediately bioavailable fraction, it actually tends to align quite nicely with the Redfield ratio. We also looked at how the composition of dust changes over elevation gradients. And we do see an increase in the organic content measured here as percent carbon and percent nitrogen with elevation. So we see an increasing fraction of organic matter as we move up in elevation. And correlated with that, we also see an increased amount of bioavailable phosphorus as we move up in elevation. With one of my students, we wanted to try to estimate what's the potential impact on freshwater systems in terms of an increase in bioavailable phosphorus based on these deposition rates that we're measuring. And so we did a very rough back of the envelope calculation for the samples we collected across the Uinta range. And here we assume that about 30% of the material that gets deposited on the catchment gets focused into a lake system. And this is an estimate that was based off our one data point, which is actually in the Wind River range, where we calculated that 30% of the dust that was deposited on a catchment ended up in a lake system. So that's only one data point. We don't actually know how this might vary in space and time, but it's the only data point we had. And so we made this back of the envelope calculation and determined that anywhere from less than 1 to about 16 micrograms per liter of phosphorus could be added to a lake system in any given year from the amount of dust that we were measuring and the bioavailable fraction of that dust in the Uinta range. And this is certainly within the range of what had been measured as an increase in the lake water concentrations through time at this particular site. And it is also within the range of being ecologically relevant. But we wanted to take this a step further. We don't really want to hand wave about this type of effect. And so I've started working with postdoc Jeff Nielsen, and we've been looking at the Green Lakes in Colorado, and we've been using SWAT, the SWAT model, the Soil, Water, and Assessment tool, where we are looking at we are using the fertilizer application module to simulate our dust phosphorus additions, and specifically looking at the readily bioavailable fraction and how that gets deposited on a catchment like the Green Lakes, and modeling it how that phosphorus or that dust material would move through that particular catchment and into the freshwater bodies. And this is the data that we used to input into the model. So you can see that from 2019 to 2021, we had a big increase in dust deposition at this particular site, but we didn't really see a huge shift in the composition of that dust. So the type of dust that we were seeing was very similar year to year, but the amount of that dust falling increased through time. And then the map that you see on the bottom is showing where we saw the biggest change in the flux of dust phosphorus through the watershed, which was really relating to the soil type across this particular catchment. So soil type seemed to play a big role in how dust nutrients were moving through the watershed and ultimately ending up in the freshwater system. And we quantified how that dust partitioning of the most soluble fraction of the nutrients ended up within plants or streams ultimately. And you can see from 2019 to 2021 that the dust fraction of phosphorus made up anywhere from 9 to 19% of the phosphorus in stream flow through those years. Didn't really contribute to nitrogen, which we anticipated because nitrogen deposition is overwhelming that signal. But nitrogen did seem to play a role in plant uptake as far as the model suggested. So this provides some evidence that the amount of dust nutrients being deposited are sufficient to alter the freshwater chemistry in these systems. But we're also really interested in doing experiments with our dust materials. So what can we see by adding dust to freshwater systems and to model ecosystems to try to understand how they might alter the ecology? And so we started off with a controlled laboratory experiment with a model organism, a green algae, and we added dust in 3 different concentrations, and we incubated this organism with that dust across 3 different temperatures and 3 different pHs. And now we wanted to set up the experiment in this way because in the real world, it's not just dust deposition that freshwater systems are experiencing. They're also experiencing changes in temperature. And with the addition of dust, you can also change the pH. And of course, there are other drivers in the environment that are altering the pH of freshwater ecosystems. So we wanted to try to understand the combined effect and independent effect of all of these different variables on the growth rate of a model organism. And so we anticipated that with adding nutrient-rich dust that we collected, that we would see an increase in the specific growth rate of this organism. But we also know that this organism and all algae have a temperature range with which they are optimal for growth. This being a very well-studied organism, we have a good idea of what that temperature range is. And similarly, there's an optimal pH range for growth for this particular organism. And with this experiment, we measured the amount of soluble reactive phosphorus that was released under each of these conditions. We measured the growth rate of the organism, and we also measured the stoichiometry of the biomass that was grown. And so these are the results for the soluble reactive phosphorus that was leached with each of these successive dust additions under different pH and temperature. And you can see as we increase dust additions, we saw an increase in the soluble reactive phosphorus as expected. And we also saw that as pH decreased, we saw an increase in the amount of soluble reactive phosphorus released. And this is what we anticipated for a couple of different reasons. Enzymes can be more effective at these lower pHs. Also, the solubilization of phosphorus from different compounds in the dust can increase with lower pHs. Looking at the effect on growth rate, again, we saw increased growth rate with each successive dust addition, but we did start to see the negative effect of high temperatures on this specific organism, which we anticipated seeing after we know that 27 degrees is above the ideal temperature range for this particular organism. And the effect of pH seemed to shift whether or not we were at relatively low temperatures versus relatively high temperatures, where At lower temperatures, we saw an increase in growth rate, which pH, whereas at the higher temperatures, we saw a decrease in growth rate with shifts in pH. But some of the most interesting things that we saw was that with increased dust addition, we saw an expansion of the temperature tolerance of this organism, as well as an expansion in the pH tolerance of this organism. So it suggests that the additional nutrients that are added from dust actually extends the range of growth, the potential growth range for this organism. So it increases the plasticity of the organism. That was really interesting. Then we wanted to understand how we could apply this more broadly. And so we wanted to try to map what might be the most sensitive areas to dust deposition, the most sensitive freshwater systems to dust deposition. So we combined known rates of dust deposition with a map based on geology of the anticipated pH of freshwater systems based on the soil pH and the soil cation exchange capacity, and combine that with the expected temperature change over the next couple of decades to create a map of which areas we might anticipate having the most sensitive freshwater systems to dust deposition. This is what the map looked like. You can see for North America, a lot of our western mountain areas have high sensitivity. The Tibetan Plateau has some of the highest sensitivity. So that was pretty interesting. So that was an experiment done with a model organism in a lab environment. So we also wanted to try to understand how can dust change a freshwater community within its actual environment. And so we carried out very similar experiments in 3 different mountain systems across the West. One was in Flathead Lake in Montana. This is a very large oligotrophic lake. Another was Castle Lake in California, a smaller mountain lake. And then again in the Loch in Colorado, which is an even smaller, more shallow lake. And so we conducted a similar type of experiment where we had many different replicates where we added low, medium, and high dust additions to lake water samples and then incubated them in situ. We also added nitrogen independently and phosphorus independently and nitrogen and phosphorus together. And we did this for 2 reasons. One was that we wanted to understand what the key limiting nutrient was at the time that we were doing our dust addition experiments. But we also added nitrogen and phosphorus at the known leached rate of the dust that we added for the maximum amount of dust. So we wanted to compare how dust nutrients might differ from raw nutrient addition within the system. And then at Castle Lake, because we had an on-site laboratory at this mountain lake, we also conducted a light and dark bottle experiment so that we could estimate net primary production. And so we incubated dust across our 3 different gradients in light bottles and dark bottles, and then to estimate production and respiration in the dark bottles so that we could get net primary production rates. And so these are the results from the 3 different lakes in terms of chlorophyll concentration, which is a good proxy for algal growth. And you can see that across our treatments, we in general saw an increase in chlorophyll A concentrations as we added more dust. And interestingly, we also did see that the dust effect seemed to have a greater influence than the equivalent nutrient addition. So we did see that dust seemed to stimulate more chlorophyll than just and nitrogen and phosphorus alone. And we think this might be because dust also adds other constituents that might be important for growth, like other macronutrients like calcium, or potentially some micronutrients. And then we also saw that through time, the slope of the growth increased with each successive dust addition. But then when we looked at our primary production data, we started to scratch our heads a little bit, because what you can see here is that the low dust treatment had the greatest net primary production rates, whereas the highest dust treatment had the least primary production rate. And so this was initially confusing to us, and is still a little bit confusing given our chlorophyll A results. And so we spent some time trying to come up with different hypotheses as to what this might be reflecting, and then testing those different ideas. And one of what we found evidence for, and what was pretty interesting, is that it seems that dust also stimulates chemoautotrophic bacteria. That's, I guess, not super surprising that a lot of bacteria would also be able to capitalize on the nutrients that are added to the freshwater system. And so we actually saw a lot of dark uptake of carbon-14, and actually we see that dark uptake of carbon-14 increases with the amount of dust that we add. Another hypothesis that we were working with is that dust adds labile DOC, not just from the organic matter within the material itself, but also because if it's stimulating a lot of primary production, it can also stimulate the extracellular release of labile DOC from that algae. And so we did use fluorescence to try to quantify that, and we did in fact see that increased concentration of labile DOC with our dust additions. So there could be both increased chemoautotrophic uptake of carbon-14 during the dark experiments, as well as increased respiration during that time. So the last experiment I'm going to talk about was this next experiment that we did, where we collected community samples from 2 different systems and then incubated them in the lab. And in this experiment, we were really interested in trying to understand how the antecedent conditions of a lake might influence its response to dust additions. And so we selected 2 different lakes that had different characteristics. So one is in a limestone basin. So the Jordanelle Reservoir here in Utah is in a limestone catchment, so it has a relatively high pH. And then we selected a lake of similar size in Wyoming that is just below the Wind River Range. So it's draining from a silicic catchment with a lower pH. And then we added 2 different types of dust. One dust material that was relatively rich in calcium carbonate, so it might have a greater impact on pH of the system. And then we added another type of dust that was relatively rich in phosphorus. And so we wanted to understand how these 2 different types of material might impact the community as well as the growth. And then we incubated those again in 2 different temperatures to simulate the effect of increasing temperatures on freshwater production. And I'll just show one little piece of this, the results of this experiment, which we found pretty interesting, is that we did see, again, an increase in the growth rate of the organisms that were present, but it was dominated by cyanobacteria. And so that was really interesting to us, because that's often a species that we don't want to see proliferating in our freshwater systems. Okay, so I wanted to talk a little bit about what we're currently working on. So some of the next steps with this project are to try to understand what's driving regional dust deposition rates. So we've collected dust deposition now monthly for many years across all of these different locations. So I'm working with some modelers to try to understand where that dust might be coming from. We also want to dig deeper into what might be controlling the seasonal and spatial distribution in bioavailable nutrients. And so we're using FTIR analysis and also visual counts of different types of material in our samples to try to understand what might be driving those shifts. I'm working with Dr. Mackenzie Skiles at the U to try to understand how different dust composition might influence dust on snow. I'm working with a microbiologist to try to understand pathogen concentrations in our samples. And then Dr. Greg Carling and Dr. Zach Andrew down in BYU have been working on collating a dust fingerprint network at the source. So collecting all kinds of different dust materials that are being eroded from different source environments, and they're creating novel fingerprints for these different dust sources. So I think it would be really great if we could take our end mix sample at our deposition sites and try to deconvolute where that dust might be coming from. And then again, I'd just like to reiterate, if there's anyone's interested in collaborating with any of these dust samples that we have, or if you have any ideas about how we might use it moving forward, I'm more than open to having conversations about that. And thank you so much for your time. And I will answer any questions.
Wonderful. Thank you, Janice. Appreciate that. It was a really fascinating talk. Do we have any questions from anybody? You can either put them in the chat or, oh, here's one. Let's see, Ling has one. Ling, do you just want to unmute yourself?
Hi, Janice. This is such an interesting talk, particularly to me. I was very excited to see those, chemical deposition from dust. So I'm like phytoplankton. My specialty is phytoplankton and harmful algal bloom, especially in the lake water, cyanobacteria bloom, taxonomy, identification, ecology, etc. So for question related to your work, so in the lake experiments, have you seen the shift of phytoplankton species over time?
Yes, so we did in both of our incubation experiments. We saw a shift in the community composition, and it seemed to vary based on the initial conditions of the lake and the initial community of the lake. So in some systems, we see a shift towards more heterotrophy. In some systems, we saw a shift towards more autotrophy. In some systems, we saw a shift towards more mixotrophic species, so organisms that can both photosynthesize as well as consume organic carbon. And then we did, in this last study that I showed, we did see the main organism that responded to dust additions were our cyanobacteria community. So that was the big increase in, I can pull that up again, actually. And the shift in the community was really driven by cyanobacteria. So that's shown here in purple with our control here, and then our phosphorus rich dust, our calcium rich dust. We did see these big increases in the cyanobacteria representation in that community.
That's really interesting. Because many years ago, I did a similar experiment, but not with dust, but with Mississippi River water and to lake water. And what we found is actually, so I actually counted the phytoplankton community under microscope. We did see the same trend with, all this just Mississippi water input. We see increasing of cyanobacteria in the lake phytoplankton community. That's great. I really would love to talk with you further, offline. And as you said, if there's any possible or chance, mutual interest for collaboration, that'd be great. For collaboration, that'd be great.
Yeah, I'd love to talk more.
Contact you more. Yeah, great.
Sounds great.
Thank you.
Ramesh is wondering, did you find iron elements in your dust sample?
Iron elements?
Right, correct.
Yes, yeah. So there's iron in our dust.
Okay. Franklin's asking, I'm wondering about carbon uptake from freshwater ecosystems. Were CO2 levels monitored in these experiments?
Can you repeat that? Sorry.
Yes, I'm wondering about carbon uptake from freshwater ecosystems. Were CO2 levels monitored in these experiments?
Oh, did we measure respiration? We didn't measure respiration with these experiments. We measured like direct carbon-14 uptake as for production, and we measured the respiration. Yeah, but we didn't measure carbon dioxide fluxes out.
Let's see, somebody had their hand raised, which I just missed. So if you have a question, I think that's all that's in the chat. If you have any other questions from anyone, just please either unmute yourself. Joe, let's see, I think you're muted, Joe. Joe, if you have a question, I think you're muted.
There we go. Okay, yeah, very nice. Thank you very much. I have a question. How would you care to extrapolate this to dust deposition to the oceans? And
Well, I think that would be tough, because what we're mainly monitoring is more regional sources of dust.
Yeah, I understand that, but I just wonder if you had any insights of the sort of experiments you might want to carry out.
Oh, yeah. Well, so our model organism that we did in the first experiment, where we measured across gradients in temperature and pH, that is a common green algae that is also found in marine environments. So we could extrapolate in that sense to what might be happening in marine systems as well, but it's only a single organism. And so it'd be interesting to carry out similar experiments under different communities, under different conditions, and some that are similar to the marine environment.
Would you consider doing such experiments? I understand you're far removed from the ocean there, but not exactly a walk to the beach, but.
Yeah, we would be very interested in doing that.
Yeah, that would be great. Great, thank you. Oh, one further question. I briefly left the room. What sort of dust were you using as your addition to your samples?
The dust samples that we collected at all of our 30 sites around the country.
Oh, okay. So you just mixed them all up and put it.
We didn't mix all of them, but for some of them we selected key sources. So for this experiment that's showing right here, we selected a dust that was relatively rich in calcium carbonate and a dust that was relatively rich in phosphorus, because we wanted to understand how those two different compositions might impact the community. For the other experiments, we selected dust across a gradient in the west so that we could sort of get a mixed dust sample, and that we could apply to each of our different experimental treatments, because we didn't want the type of dust to then influence our results. So we used the same dust in all 3 mountain lake systems.
Good. Thank you.
Okay, this next question is from Santiago. I wonder, did you consider differences in the state of weathering in the dust? For example, I would expect that dust on snow that eventually reaches water streams via melting may have processed distinctly than dust blown from a playa.
Yeah, absolutely. I think that dust that has been sitting on snow for a while and has experienced the melting of snow would probably leach some of those nutrients. But we didn't collect dust on snow, so we don't know how that compositionally might compare to what we capture in our samples, but we would just anticipate that it might. And that's one of the reasons why we wanted to use the NADP network and the sampler that we have, is so that we wouldn't lose any of those bioavailable nutrients to leaching by having to use water in our sample to collect that dust material. So we could just scrape it directly off the glass plate with a razor blade and then subject it to our different experimental treatments.
Virginia's asking, any idea why the SRP increased from 2019 through 2021 in the SWOT slide earlier in the talk? Is it from fires?
The amount of dust increased, and that was pretty true broadly across the West. We saw from 2019 to 2021 an increase to 2022, actually, we saw an increase in dust emissions and deposition across most of our sites. This year, we saw a dip again, and I assume that has to do with climate and the factors that lead to the generation of dust. I did compare it to aridity indices as well as wind speeds, and I saw nice correlations, but that's hard over a 4 or 5 year period, and it's a little bit out of my range of expertise. So that's one of the things where I would love to collaborate with somebody on how we might tease apart what's driving those increases in dust deposition through our sampling period.
Tom Gill's wondering, tell us more about the tire-related materials you found in some samples. Were they from any particular sites near highways or remote sites? What are the possible ecological implications of any tire materials getting into waters, etc.?
Yeah, tires are a little bit harder to quantify using FTIR. So we've done a little bit of both FTIR and visual counts of tire wear particles. We find them everywhere, even in our really remote locations. They are, in terms of by number, they're about 50% of what we saw in terms of total polymers. And in terms of what their impact might be, they're very dark. They do absorb a lot of radiation from the tests that Mackenzie and I have done. And it's, as I'm sure you guys might be aware, there's been a lot of really interesting studies about how tire wear specifically leaches certain chemicals into freshwater systems that's really toxic to salmonids. So it's possible that those tire wear particles are moving those contaminants to remote locations, but whether or not that's in any concentration that is ecologically relevant, I have no idea. And trying to isolate samples that small is also very, very difficult, since they're generally about 100 microns long and about 10 or 20 microns wide. It's really difficult to figure out how we would physically isolate them to test anything like that.
Just a couple of hands up. Cassie, do you have a question?
I do. First, always a pleasure to hear you speak, Janice. Really nice talk.
Thank you.
My question for you is, are you planning on looking at how some of your experiments, in particular on this slide, might impact toxin production? Like, have you thought about looking at maybe if the dust is also playing some role in increasing the ability of some of the cyanobacteria to produce some of the toxins you might find in these freshwater systems?
Yeah, so we are just starting a study in Wyoming. Some of those alpine areas this year and last year have been experiencing toxic cyanobacteria blooms. Mm-hmm. And what's interesting is it seems to be happening in sites that we know have historically had a lot of dust deposition, and the sediments have high phosphorus concentrations across that gradient in dust deposition. And maybe what's unique about the last 2 years is how warm the fall has been. And so we're setting up some experiments to try to understand the relationship between the sediment phosphorus and how that we want to try to quantify how that might have come from dust, or what fraction has come from dust, and how combined with temperature that might be leading to these cyanobacteria blooms. And then again, how that might be impacting the toxin concentrations in these systems, which the Forest Service is really concerned about, because you can't filter cyanotoxins out of water, and these are areas where we don't anticipate seeing those kinds of things. So yeah, that's a super interesting question, and we're still trying to wrap our heads around exactly how to test that and how to how we're going to set up the study.
Yeah, I actually had a student who did a senior thesis project looking at the role of temperature, just temperature alone, on HABs and toxin production. I mean, we didn't get super far into it, but maybe we can chat offline about some things that might be helpful for getting your experiment going, because I think it's so important with how much of a problem these HABs have been around the country.
Yeah, I'd love to.
Thanks.
Thank you.
Bernie, you have your hands up.
Hi, yes, thanks for a great presentation. And getting back to the dry deposition, how are you still doing that? And how frequent are those measurements?
Yes, we're still doing it. We're measuring monthly at each site. And I anticipate, well, we have enough money to do it until next summer. I'm going to try to apply for more funding to carry it long term, which would be great, but who knows? But yeah, we are still collecting from about 25 sites.
Okay, because on a month-to-month basis, you might be able to monitor with satellite. And I see that Bill Lyon is going to be giving a presentation in a couple of weeks. A lot of times with satellite imagery, we can see big dust events, even smaller dust events, and that might help give you an idea of pinpointing where some of the dust is coming from.
Yeah.
So,
that would be...
yeah, because that's very interesting, and I often wonder, how, even from satellite, how do we verify that what we're seeing is blowing dust, and where is it deposited? Yeah. So, okay,
yeah,
great.
I would love to compare notes on something like that.
Okay. Thanks.
Thank you.
Did I miss any other questions? It looked like there was one here that went away, but if I missed a question, feel free to pop up. We might have a minute or so left before we should wrap up. Is there anyone else? Astrea is asking, what is your sampling method? Not sure, Astrea, if you want to hop on and be specific about
sorry. Yeah, just the instrumentation that you're using. I'm sorry.
Yeah, so we're working through the NADP network where they use dry and wet deposition samplers so that you can capture what falls with rain independent of what is falling out dry. And so we developed a sampler that fits into the dry bucket, and now I'm scrolling back endlessly to try to get to that slide. Maybe I'll just find it. So this is what a typical NADP site looks like. And they have this dry and wet sampler bucket, which is triggered by precipitation. So you can independently capture dry fall and wet fall. And then we put the sampler in the bucket, we sterilize it. It's got a glass plate at the bottom and then 3 different size screens. And the screens are really good at, trapping any bugs that might fall in there so that we can keep them separate from the sample. And then we take that glass plate and we use a ceramic razor blade where everything is sterilized, UV sterilized or with ethanol. And then we scrape that dust off the glass plate, which was an idea we got from movies. And then we are able to capture that dust material dry and outside of a filter, so we can actually take that dust and analyze it without interference from a filter. And we can also add it to our freshwater samples or our experimental system so that we can see the impact of that dust.